Hello and welcome to the recording of our webinar that we gave on January 27th, 2021, on the new Lifelines data platform and more specifically the data model. My name is Trynke de Jong. I'm a project manager at Lifelines, and we organized this webinar together with my colleague Laura, who is a data manager at Lifelines. The scope of today's talk is the platform, but more specifically our new metadata model, which we implemented on that platform. And this is the thing, so to speak, which many of you researchers will work with and will notice changes. I will very briefly introduce the Lifelines dataset. Many of you will know us already. For those who don't, I will very briefly tell you something about Lifelines and more specifically why our data model is such a challenge to complete. Then I will introduce the new metadata model, especially why we changed it from our old data model, which many of you are familiar with. I will introduce some new terminology, which is good to know in communication between Lifelines and users. I will show you briefly the new data catalog and the Wiki. I will explain the structure of release tables, which is quite different from what it was before. And I will tell you something about the things we still have to do in this context. I will share 5 take-home messages. And what I will not talk about in detail is data linkage, because that's the topic of our webinar on March 4th. I will give that as well. And I will not talk in detail about omics, genomics, microbiomics, etc. That will be the topic of our webinar on March 30th. Okay, so very brief introduction of Lifelines itself. The most important thing for you to know is our mission. We want to make data and samples from our cohort available for research in the field of healthy aging. That's our mission. So we do not use the data and samples for our own purposes, for our own research. The main mission is to share it with you. And that's important because it means that we really want to invest in making this data and these samples easy to use and to maintain a clear data quality and a clear data model that is really built for sharing. We collect a lot of data and samples. We do that in the context of our general assessments, starting with the baseline assessment, which was between 2007 and 2013. We have some follow-up questionnaires. Then we had our second assessments between 2014 and 17, and we're currently running our third assessment. And in these assessments, we really try to collect the most basic of health and lifestyle related information and body physical measurements and some basic body samples such as blood and urine. We also allow additional assessments. So this means that we have an open protocol and we allow researchers to initiate additional assessments based on their own very specific needs for their research question. And this can go from really big and broad, so the whole population, for example, all the adults receiving a questionnaire or the whole adult population being genotyped, to very small and precise, like a very small subset of people, for example, receiving an eye exam or a CT scan. These additional assessments can be anything. We really make use of various collection methods, and this data is then integrated with the existing database. So for example, we have imaging studies, eye exams, all kinds of omics, questionnaires, pedometers, we've had diaries, app data, all kinds of things. And this data in all its shape and form and sizes and in all large and small subcohorts must be integrated, which is quite a challenge. We want to do this rapidly if the situation arises, and of course now we very clearly are in such a situation. COVID-19 is among us, and we set up an additional assessment with repeating questionnaires or bi-weekly or 4-weekly questionnaires. And luckily for us, we had our new systems and our new model in place so that we were able to really rapidly collect the data, implement it in our data model, and release it in new standards. We also release secondary and linked data. So secondary data is data that is derived from primary data that we collected directly from our participants. So you can think of results from sample analysis. You can think of codes. So we ask a lot of open text questions like what type of cancer have you suffered from? And then we code this open text field into something that you can use for your analysis. Some scores, all kinds of calculations. We have quite a few diagnostic instruments among our questionnaires, and the answers to these questions must be calculated to get a diagnosis, for example. So these type of secondary data we also want to import and integrate in our database and release to you to be used. We also allow data linkage. We do that, for example, via pseudonymization of identifiers. So both data sources do that in the same way, and then you can connect the data together. Or we link not via persons, but via, for example, home addresses. So that could be environmental data, for example. So the point that I'm trying to make here is that it's a very varied and very diverse and very large and complex data set that we need to manage. And up to this point, what we did was we organized the data and put it into tables. That's what we did as Lifeline. So we decided, okay, which data goes into which table. And sometimes that was very logical, there's no doubt about it, but we also have quite a bit of data that is difficult to find the correct table for, let's put it that way. And then researchers or users could order these tables, so they would go to the catalog, they would pick out the tables that would be of their interest, and then these tables would be pseudonymized and released to the researcher. And so of course these tables are not standalone tables, they're interconnected. There is a data model underneath it which integrates all these tables together, and basically it looks like this, the picture of that interconnection, or this spider web of tables. And you can see that it looks quite complicated, and that's true. And anytime there is new data that must be fit into this system, it's really complex to get it at the right spot and fully integrated with everything else. So the type of problems that we had, so the feedback that we received from mostly researchers, first of all, it was difficult to promise or to make a date where new data would be released. It would always take a lot of time to get the data ready for release, and so we would always have to postpone, and would be very unclear when research could really expect new data, which is of course not very handy. The data in a workspace is different from what I expected, is what we hear a lot, because you think maybe that you will get 160,000 data points for each variable, but of course that's not the case. We have many variables that are only collected in a certain subcohort or at a certain time point. And so it would be quite difficult to communicate with users what it is exactly that you will get. And you really need to, first of all, ask the right questions, and data managers would really have to find the right answers to give, and it's difficult to get to really get to the bottom of this. So that's difficult. We hear that it was very difficult to organize the data. So you get it, of course, in certain tables, but then you probably want to reorganize it for your own purposes, and the way that it was released would make it that very time-consuming and very complex. It's difficult. Many of you do research which depends on longitudinal data, so the same data collected over time, and it's sometimes quite difficult to recognize the data, to find it, and to connect it in a syntax in a certain way. So that was difficult. There was not a very clear policy on metadata. So some metadata was overcomplete, some was missing, and it would be quite difficult to standardize that, or it was not very well standardized. the data or information is missing on who was invited for an assessment. So you would know who provided answers or data points, and you would know who did not provide any answers or data points, but you don't know of the people who did not provide, you don't know who was actually invited and who was never approached at all. And that's a relevant distinction because you might want to take that into account in your bias analysis of responders versus non-responders. So that data was missing. Other types of empty cells. So of course there could be many reasons why a cell in a table is empty, and the backstory of that emptiness was really a puzzle to figure out. Yeah, we collect certain variables for a really long time, and it's very possible that sometime in that period we change the protocol, and we have the instruments and the description of the instruments, but it could sometimes be really difficult to figure out. Okay, but what how is that connected to my data? And so that was an issue. There's a lot of sum scores out there that we know exist, so that we know, and of course researchers hear about this, that certain really nice complex sum scores have been generated. So of course it would be really nice to share these, to not do all the work over again. And yes, up to now these sum scores are really only available in a limited way. So yeah, that was an issue. And we also heard quite often like, hey, I noticed there was an issue, a quality issue, I reported it and nothing was done with it. Why is nothing done with it? And so from the data manager's point of view, there are also some problems. So it's not for nothing that it takes a long time to release data, because it's really a lot of work to get it properly done, really weeks, but really more months, and even in some cases years to get it really good. And by the time you're done with it, you know that you can start over again because new data has accumulated in the meantime. So that was really, time consuming. A lot of data is therefore on the shelf to be released. We have it, it's nice, you can use it for your research, but it's not ready to be released, and it's kept on the shelf until we find the time to really integrate it in the data set. The choice where to put data is in the hands of data managers, and it could be problematic. So some data is really easy to distribute, but other data is really difficult to classify. Like, where should I put it? I can put it here, but a lot of you might really want to see it in another table. So how do I do this in a way that's workable for everybody? Once data is in the release, it would still be quite time consuming to prepare a data set for a specific user. We would really get a lot of repetitive questions. Of course, for researchers, it's always the first time, but data managers and also project managers, we really got used to answering the same questions over and over again. And we really thought, okay, there must be a better way to communicate. Users get a lot more data, and that's first of all not very handy from the user's point of view, but also from the general principle of data minimalization point of view. So the GDPR really states, okay, personal data should be minimized when it's shared with other people, and so we really wanted to make data sets for release more compact. Based on this complex structure, we would really spend a lot of time on supporting researchers in the workspace. For us, or for data management, it was also a problem to really organize metadata in a clear way, that you would not have to think about it every time, like, okay, how am I going to deal with this metadata, but that there is a clear policy on it. And repairing mistakes in this spider web of tables is really difficult and time consuming, and often would just not be done, and would say, okay, we'll fix that in the next release. And sometimes that takes a long time. So our solution to many of these problems is to increase the resolution of our data. And basically that means that instead of storing our data in tables, we want to store it in data units, so to speak. And these data units can then, for a specific release to a specific researcher, be then organized again in tables, so that it would be really specifically those data units that a person needs. So what do I mean with a data unit or a data point? So that's this little Lego block with a number in the middle. It could also be a piece of text, but that 1 and that 32.2, that's the actual data that you use to calculate. And then there's a sort of like a shell of basic metadata around each of these data points. And so there's who, what, and when. And who meaning, of course, the participant that delivered that particular data point, and that participant has certain specific characteristics that are really stuck or matching that particular person. So they have a participant ID, and when you release the data, of course, they are then receiving a specific pseudonym. They have a sex, they have a date of birth, they have a postal code, and they could be a member of a subcohort. The what is the variable that is collected with a variable name and a label, and sometimes, let's say, an answering, an enumeration, we call that, so an answering option. And when is the context in which that data point is collected. So that is a date, so there's an actual absolute time point where it was collected, but it was also collected in the context of a specific assessment. And there is some new terminology there that I will introduce to you in a minute, but that's the basic principle. And you can do that for each of our available data points. So we did that. So 32.2 is a boy, a young boy in the context of a visit when his body weight was measured. And so each data point comes with a set of very limited but very crucial basic metadata. Nothing extra, anything extra. So there's of course a lot more metadata to be shared, but that will be provided on an aggregated level, for example, in the Wikipedia and in protocols. So that's more aggregated. This particular metadata, basic metadata, will be provided for each and every single data point. So how do we organize then these data points and this metadata? So we have a long list on our data platform with participants and their basic characteristics. And we have a long list of variables and their basic characteristics. And we have a long list of assessments with their elements and variants. And that I will briefly explain. So an assessment is an overarching data or sample collection with a common goal. And this is, for example, baseline measurement is an assessment, 2A, follow-up questionnaire 1B is an assessment, but also an additional assessment. Let's say, for example, an additional questionnaire will have an assessment name, and more complicated things like, let's say, email live with lots of measurements as well. And within an assessment, you may have one or more elements. And the basic definition of an element is that it's a part of an assessment that takes place on one day. So each data within an element will have the same timestamp for a participant. Within those elements, you can recognize variants, and sometimes there's only one, sometimes there are 20, it's all possible, but the definition of a variant is that it's a very highly standardized set of variables under a very specific protocol. And what I mean with protocol is that it has a very specific participant selection, so they adhere to specific criteria, for example, 18 plus or female, or a combination of many criteria. That's all possible. A variant has a fixed set of variables, so if any variable changes or is dropped or is replaced, then you get a new variant. And of course, a variant is measured or collected under a very strict collection method. So if anything changes there, you will get a new variant. And an example is that if you go from a paper questionnaire to a digital questionnaire, even though the set of variables is 100% the same, the participant selection is 100% the same, but because you go from paper to digital, it's a new collection method, and that's a new variant. So any changes, you will get a new variant. And so what we do then is we have also on our platform a complete cross table of which participants participated in which variant. So a full list of all the who, when combinations, variants in this case. We also have a cross table with a full list of combined when and what. In other words, which variant contains which variable set. So these are two important cross tables. And the third cross table is the combination of both cross tables, let's say, and there you can call it the who, when, what. And that's 725 million unique records or data points, if you will. So what happens now with this information, so it's on the data platform, including the data, but the relevant metadata, so the relevant metadata of these, specifically these cross tables, is then every night fed into our new data catalog. And this metadata is organized then over participants and assessments and variables. So you can recognize the basic metadata structure in our catalog. So we have information on the persons, and you can use that information to filter your participant selection. Not everything, so we only offer limited filter options. We could add to them if we want to, but for now these are the limited options to filter participants, but it will in many cases help you to really reach a compact data set. We allow you to filter assessments. We're not going to filter on variants. You can do that once you have your data in your workspace. You can maybe discard some variants because they don't fit your research needs. But in principle, you can say which assessments, so at the level of assessments, which one do I want, which one do I not want, and which one am I not allowed to get. For example, the COVID or COVQ, which you see here. If you didn't get specific approval for that data set, you can't, or you're not allowed to order it. You can try, but it will be discarded. So you can filter out the assessments that you don't need. And of course, then you can, as a third step, you can select the variables that you need, and you can select it per variable. So you really can narrow down what it is that you want in your data set. So anything you select here will be translated into a so-called JSON, and that's the message that is then being sent to the platform, and the platform can automatically pick that up and generate tables. Just a brief thing that I want to say here. So you will notice in the catalog sometimes little information circles or buttons. And if you click on those, you will be sent to the Wiki page. And this is to get more general overarching information about a specific time point or a specific participant filter, for example. So just to briefly introduce the wiki, so this is what it looks like. It's a very large collection of wiki pages where it is precisely described how our protocols work, what kind of data do we have. You will be shown, okay, if you're interested in this data, then maybe you want to have that data as well. And very importantly, you can make use of the search bar, which is maybe a little bit hidden on the page. You may not see it immediately, but if you are looking for any information on anything in particular, please use the search bar. But in principle, all the information that you need is shown, and more importantly, I think, connected to related information. So you will see for a specific data, okay, when was it measured, and what does it mean that it was measured in 2A, and what else was measured in 2A, and how much time was there between 2A and 1A, and all kinds of sort of general overarching protocol information can be found on the wiki. We are continuously updating it, and we would be extremely grateful for any suggestions here. So we're really trying to make this an interactive thing where you can really request changes or additions, or really want you to help us complete this as much as we can. Okay, so the JSON that I told you that was sent from the catalog to the platform basically triggers the automatical generation of tables. It will also automatically pseudonymize these tables. And this is what it looks like when you go into your workspace. This is where you find the results of that table generation. And yeah, you will notice, or now you understand probably, that we release these tables to you per element. So the element is the defining factor which decides which data goes into which table. So that looks like this. So anything before underscore results, that's the name of the element. So 1AQ1 means assessment 1A, questionnaire 1, and that's one of the elements of baseline. We give you a few additional tables. So one will contain variable metadata, and this is where, for example, you will find the new variable name and its equivalent old variable name, the historic variable name. And of course, we know that this can be crucial for researchers who have been working with our data before and built their syntaxes and their knowledge of our data set based on these old variable names. So you can definitely find them and make use of them. We will give some information on enumerations. This is the new term for our answer codes in questionnaires. So 1 meaning yes, 2 meaning no, that's enumerations. And we will give you a global summary of containing participant information, so date of birth, date of death, more importantly, intervals between elements. So each participant will have participated in certain elements, and the time between them will be given. And which is not yet complete but will be completed is which participants were invited for all the elements in your data set. Right. An important aspect that I would want to introduce are the missings. These are the empty cells that I talked about before. So what does it mean that a cell in your table is empty? And what we did was, so now you know more about the what, when, who system. So let's say that a participant participated in a variant, and that variant contains a variable, yes. And that participant also delivered information or data on that variable, yes. Then clearly your cell will have contents, meaning data. If your participant participated in a variant and that variant contains a variable, but that participant did not deliver that variable, then you will get a missing token. And that basically means the participant was given the opportunity, let's say, to provide the data, but failed to do so. Maybe because he or she skipped the question or refused to participate anymore, something like that. The third option is that a participant indeed participated in a variant, but that variant did not contain that particular variable. Therefore, the participant could not have delivered the variable, and in that case, the cell is empty. So if you see that, it means your participant was never in the situation that this variable was collected for that time point. If a participant did not participate in a variant that collected that variable and could not have delivered the variable for that purpose, then there is no cell at all in your table, so you will not see any information for that participant and that variable. And that's relevant, for example, in this particular case, there's many of these where we ask, okay, did you have a certain disease or not? In this case, agoraphobia. And well, some people say yes, some people say no. A lot of people skip the question, but they've seen it. And so skipping it, although we cannot guarantee that it means no, it's quite likely that it means no. They skipped it because they don't have the disease and they didn't take the trouble to say no. There will also be a lot of people who did not ever get this particular question, and that information, those empty cells, we have no idea whether they developed agoraphobia. So it could either be yes or no. So in your analysis and your bias control and your percentage calculations, you can really be more precise now, we think. Okay, our to-do list. We really want to complete all these short, brief, renewed protocols for each variant. We are well underway, but we definitely miss some still, so we're working on that. We have not yet annotated our questionnaire PDFs with the new code, so you would still, if you look at the PDFs, you'd still get the old code, and then you would have to go to the table with the translation to the new variable name, and we want to improve on that. We have quite a bit of additional assessment data that we still have to import. Also some secondary and linked data sets that we have to import. Again, we are working on that. We have quite a few secondary data already imported, so you can find them in the catalog, but there are still some on the shelf that we want to get to. We continuously update our wiki, and yeah, we hope actually that this webinar will really urge you to give us feedback, to report mistakes or problems, and that we will get to fix. Okay, so that gives me again, or that gives me the first take-home message, which is really the question for feedback. So you are our main source of information on this. You are using this stuff. So please, if you notice anything or something is annoying or frustrating or wrong or unclear, let us know. Obviously, we're also welcoming positive feedback, so please don't forget that. If you're very happy with how we do it, let us know as well. But of course, yeah, the feedback about problems are more important. That does not mean that when we receive that feedback, we will immediately or ever fix it. We will usually might have to make compromises depending on who is asking and for what purpose, and can we really make the change without affecting other people's work, because sometimes that's the case. What works for you may not work very well for somebody else, so we sometimes have to balance that out, but we will explain to you how we do that, or if we do that. We really want to urge you to start your project before you get to the workspace, so to really make use of the wiki and the catalog to figure out what it is that we will offer, what you will see in the workspace at the end, get to know the structure, the principles, the terms, what to expect. Spend some time doing that, and any time you put in there on the outside, you will earn it back in the workspace, because you will be able to much quicker go into the workspace and understand how and where to connect your variables in your syntax. Do that with your students. If I don't know who's listening to this, but if there are any supervisors or group leaders or professors who need to do a lot of teaching, make use of it. The catalog and wiki I think are excellent teaching materials for students who are in the future maybe will be doing some of this type of research. Make use of it and think about, okay, how do I set up my project? What do I need? What do I need to fix once I get to the workspace? Our data set is not perfect, so some of the things that are missing or are imperfect, like how am I going to deal with this once I'm in the workspace? Read the manuals, that's clear. We spend quite a bit of time answering questions that are really written out carefully in the manual, so we really ask you or advise you to read it. If whoever is listening is working currently on a data set in a workspace, an old data set in an active workspace, then you're eligible to renew it. So please ask our data managers if you're interested to renew your old data set for your analysis. And the last take-home message, because we notice this is going wrong, our catalog is very inviting. The COVID data is very easy to order. You can click on a button and it's in your basket, but this COVID data is really not available for anybody. You really have to get approval, specific approval, so please don't order it if you don't have that approval, because it means that you might have to do your data order again, so be aware of that. Okay, well, this is the end. Normally we would start a live discussion now. That's not possible, but obviously if you have any questions or feedback or any other information for us, let us know via email. I've shown the addresses here. So let us know, and thank you very much for your attention. Thank you.